Photoactive stomata. White circle stomata that open during the day and close during the night. Fig. 6.3 Structure of stomata. Scotoactive stomata. White circle of stomata that open during night and remain closed during day. Guard cells are bean-shaped in dicots and dumbbell-shaped in monocots. The concave wall of the guard cell towards the pore is thick and elastic. As the turgidity in guard cells increases, the thinner wall gets stretched more as compared to thicker wall. This helps in the opening of the stoma. Guard cells also have radially arranged cellulosic microfilaments, which also help in opening of stomatal aperture. Types of stomata. Anomacytic type. Stomata are surrounded by cells and do not differ from other cells. Parasitic type. Stomata by two or more subsidiary cells which lie parallel to the long axis of the pore in guard cells. Anastocytic type. Guard cells surrounded by three subsidiary cells in which one is smaller than the other two. Diacitic type. Guard cells surrounded by two subsidiary cells whose common wall is at a right angle to the long axis of pore in guard cells. Actinocytic type. Surrounded by four or more subsidiary cells which are elongated radially to stomata. Fig. 6.4 Stomatal types. A. Anomacytic. B. Anastocytic. Circa. Diacitic. D. Parasitic. E. Actinocytic. F. Cyclocytic. G. Tetracytic. Fig. 6.5 Stomatal types. A. Lipocytic. B. Parasitic. Circa. Desmocytic. D. Polycytic. E. Sporocytic. Distribution of stomata on leaves. Apple and mulberry type. The stomata are found on the undersurface of leaves only. Potato type. When stomata are more on undersurface than the upper side, they represent the potato type. Oak type. Stomata are equally distributed on both surfaces. Water lily type. In such cases, stomata are found only on the upper surface. Potamogeton type. It is represented mostly in submerged aquatic plants. Stomata are either absent or functionless in such cases. Daily movement of stomata. Lockfield classified stomata based on their daily movement into four main types. Alfalfa type. In this type, stomata remain open throughout the day and closed all night. Examples: radish, mustard, turnip, pea, and apple. Potato type. Stomata remain open throughout the day and night except for a few hours following sunset. Examples: cabbage, pumpkin, and onion. Barley type. Stomata remain closed throughout the night and open only for a few hours during the daytime. Examples: barley, maize, wheat, and oat. Acquisitum type. Here, the stomata never close. Example: acquisitum. Opening and closing of stomata. Opening and closing of stomata. Potassium ion theory. Levitt, 1974. The opening and closing of stomata are controlled by the accumulation of solutes in the guard cells. The guard cells actively take K plus ions from the neighboring subsidiary cells. Figure 6.5. Influx of K plus during the opening of stomata was observed by Gugio, 1967. As a result, the water potential in the guard cells decreases and water molecules move from higher water potential subsidiary cells to lower water potential guard cells. Due to endosmosis, passive guard cells swell, become turgid, and the stomata open. When stomata have to close, the ions are actively pumped out of guard cells. This results in the decline of solute concentration in the guard cells and water moves out, exosmosis, making guard cells flaccid. The major ion taken is K plus. Accumulation of large amounts of K plus ions is electrically balanced by chloride ions. Maleic ion accumulation also helps in the opening of stomata. Maleic ions are formed by hydrolysis of starch. Fig. 6.6 Opening and closing of stomata. Starch sugar hypothesis. During the night as the stomata close, CO2 produced by respiration accumulates and reacts with water to form carbonic acid, which dissociates to give H plus and HCO3. Ions. Accumulation of H ions lowers the pH acidic, which favors the conversion of glucose into starch. This decreases the solute concentration and exosmosis takes place from guard cells resulting in closure of stomata. During the day, CO2 is consumed, which results in an increase in pH basic, which favors starch to sugar conversion. So, during the day, starch gets converted into sugar in the guard cells, which increases the solute concentration and results in endosmosis and opening of stomata. Factors affecting stomatal opening light. Stomata open in light and closed in the dark. The light intensity required to open stomata is very low. In cam plants with cresylation acid metabolism, stomata open in the dark and closed in light. Examples: pineapple and agave. Temperature. Stomata remain open at a range of moderate temperature. At low and above 30 degrees Celsius, there is a decline in the stomatal opening. Water availability. If less water is available to the plants, stomata tend to remain closed. If available water is less and transpiration is high, the plant undergoes water stress or water deficit or moisture deficit. CO2 concentration. With increased CO2 concentration in the leaf, stomata close. This may happen even in the presence of light. Diffusion pressure deficit (DPD). Pure water contains maximum diffusion pressure. It gets lowered by adding solute to it. The difference in diffusion pressure of pure water and water in a specific solution is called the DPD of that solution. At full turgidity, DPD equals 0. In non-turgid cell, DPD equals op WP or TP. DPD determines the direction of water movement when two cells or solutions are adjacent to each other. The direction of flow of water is always from the region of lower DPD to higher DPD. Example: cell A has op equals 10 and TP equals 6. DPD of cell A equals op TP equals 10 6 equals 4. Cell B has op equals 10 and WP equals 4. DPD of cell B equals op WP TP equals 10 to 4 equals 6. So, the movement of water will be from cell A to BIE, from lower DPD to higher DPD. Antitranspirants. These are the chemical substances which can minimize the rate of transpiration. These are sprayed over the crop plant to minimize the rate of transpiration without affecting the uplift of carbon dioxide. Antitranspirant is necessary to check the wilting as well as to prevent poor yields. Examples: white circle phenylmercuric acetate (PMA), abscisic acid (ABA), and acetylsalicylic acid (aspirin) are the metabolic inhibitors which are used largely to minimize the stomatal opening and finally transpiration. White circle silicon emulsions, plastic films, and wax emulsions are used in the form of film to cover the surface of the leaf in order to reduce transpiration. Wilting. It is a state of flaccid condition of shoots and leaves when the rate of transpiration exceeds the rate of absorption of water. Incipient wilting, partial loss of turgidity, which does not cause visible wilting. Temporary wilting. During summers, when plants undergo excessive transpiration, the leaves and stems droop down. At night, the transpiration decreases and the cells regain turgidity. Permanent wilting. This occurs due to a deficiency of water in the soil and the wilted plant cannot recover. This type of wilting results in death of the plant. Permanent wilting percent. The percentage of water in the soil at which the plant undergoes permanent wilting, it is 1% to 15%. Bleeding. It is the exudation of the cell sap from injured parts of plants under the influence of root pressure. Under high root pressure, the plants growing in moist soil start oozing out xylem sap from the injured region of the plant. Example: the secretion of milky latex from the latex ducts in some plants. Uptake of minerals. Mineral absorption is mainly active and very less passive. Active absorption is required for two reasons: white circle minerals are present as ions in soil and the plasma membrane does not allow the entry of charged particles. White circle, the concentration of mineral ions in the soil is very less. Specific proteins in the membrane of epidermal cells actively pump some ions from soil to the cytoplasm. This needs ATP. Transport proteins of endodermal cells are the checkpoints where the quantity and type of solute that reaches the xylem is checked. Cells of root endodermis have the ability of actively transporting the ions only in one direction. Translocation of mineral. Minerals are translocated along with water. The sinks for minerals are meristems, young leaves, developing seeds, fruits, and storage organs. Unloading of mineral ions takes place at fine vein endings by diffusion or active uptake. Elements like phosphorus, sulfur, nitrogen, and potassium are remobilized from older to younger part. Elements like calcium, which are the structural components, cannot be remobilized. Analysis of xylem exudate shows that most of the nitrogen is carried as organic compounds, amino acids, and very less as inorganic ions. Small amounts of P and S are carried as organic compounds. Translocation of food through phloem. Food sucrose is transported through the phloem from source, site of synthesis, to sink, site of storage or utilization. Normally leaf acts as a source and storage organ. Root in case of radish acts as a sink. Source and sink may change according to season. For example, during spring, when buds are growing, buds act as a sink. Roots supply the food, acts as source. As the source sink change, the direction of movement of food in the phloem is bidirectional. Carbohydrate is synthesized as glucose and transported as sucrose. Sucrose is actively taken by companion cells and actively loaded into sieve tubes. As sucrose accumulates in sieve tubes, solute potential Y increases and so water potential psi W decreases and water from the surrounding moves into the sieve tube. This increases pressure potential and so sucrose moves from this sieve tube to another sieve tube with lower pressure. Sucrose is actively loaded into the storage organ's sink. As sucrose level decreases, solute potential decreases and water potential increases. This results in the movement of water out of the sieve tube, which lowers the pressure. Important information. Root pressure is absent in gymnosperms. Imbibition pressure in P is 1000 atms. Cohesive force is called as the tensile strength of water. Stomata remain open at relative humidity above 70% and close to low relative humidity of 50%. The term digitation was coined by Berger Stein, 1887. In Saxifraga, the rate of digitation is high during flowering. Plants growing at high altitudes show xeromorphy, a type of adaptation to reduce the rate of transpiration. Blue light of visible spectrum promotes stomatal opening. Psychrometer is used for measuring relative humidity as well as transpiration. Hydrometer is used for measuring the density or specific gravity of a liquid. Tensiometer is used for measuring soil water tension. Manometer is used for measuring pressure like root pressure. Barometer is used for measuring atmospheric pressure. Barograph represents the recording of a barometer. Porometer is used for measuring the size of stomata. Atmometer is used for measuring pull caused by evaporation of water from a porous pot. Photometer is used for measuring the rate of transpiration. Pressure chamber is used for measuring the water potential of whole leaves, shoot, etc. Cryoscopic osmometer measures the osmotic potential of a solution by measuring its freezing point. The apoplast is a non-living continuity i.e. through intercellular spaces. Symplast is a living continuity i.e. movement of water through plasmodesmata. Cells, when treated with auxin, can absorb water even from hypertonic solution by an active process. The amount of water left in the soil after the plant has permanently wilted is called wilted coefficient. Stomatal index, percentage number of stomata as compared to all epidermal cells, including stomata, in a unit area of the leaf. Salisbury gave a correlation between the number of stomata and epidermal cells per unit area. Where I equals stomatal index, S equals number of stomata per unit area, E equals number of epidermal cells per unit area. Stomatal frequency, number of stomata per unit area of leaf. Physically dry soil, when there is no water in the soil. Physiologically dry soil, when the soil contains water, but it is not available for the plant. For example, in saline marshes. Pollard, total amount of water in the soil, Crissard plus Etchard. Crissard, available water for the plant, capillary water. Etchard, non-available water, combined water is water of crystallization. Hygroscopic water is absorbed water on soil particles, and gravitational water. Chapter summary. In plants, the substances can be transported over short distances, cytoplasmic streaming and diffusion, and long distances, translocation. Transport can be of an active and passive type. Pure water has the greatest water potential. Osmosis is the movement of water across a semi-permeable membrane from its higher to lower concentration until equilibrium is achieved. Imbibition is a type of osmosis in which an absorbent is required. Introduction. Plants produce a wide range of chemicals categorized into primary and secondary metabolites. Secondary metabolites are different from primary metabolites, e.g., proteins, lipids, carbohydrates, and nucleic acids because of their restricted distribution in various groups of plants. Recent studies have shown that the expression of secondary metabolites can be modified by advanced molecular techniques. Secondary metabolites such as terpenoids, phenolics, and alkaloids play defensive roles in plants and also act as modulators in response to abiotic stress. Secondary metabolites lie at the interface between primary metabolism and the interactions of organisms with their external environment. Figure 7.1. Secondary metabolites. Secondary metabolites include pigments, alkaloids, antibiotics, nucleosides, peptides, lectins, terpenoids, antibiotics, essential oils, and drugs. Figure 7.2. Organic compounds produced by bacteria, fungi, or plants, not directly involved in reproduction, development, and normal growth of the organism. Produced in small quantities, so their extraction from the plant is difficult. Formed during stationary phase, also known as idiophase. Figure 7.3. Serving as antibiotics and producing pigments. Unique in different plant species. Active against foreign invaders, might be involved as a defense mechanism. 7 secondary metabolites. Figure 7.1 flowchart of secondary metabolite production. Figure 7.2 broad classification of plant secondary metabolites. Fig. 7.3 schematic representation of the biosynthesis of secondary metabolites. Terpenes, terpenoids. Largest group of secondary metabolites, normally insoluble in water. Synthesized either from acetyl-CoA or intermediates of glycolysis. Polymers of isoprene 1 units consist of branch 5 carbon isoprene. Often referred as isoprenoid compounds. Figure 7.4. Terpenoids are built from repetitive 5C structural motifs called prenyl groups. Grouped by the number of 5C units, have in their skeleton. Isoprene is also a principal reactant in the formation of ozone in the troposphere. 2 biosynthetic pathways. These are MBA, mevalonic acid, and methyl phosphate pathway. Figure 7.5. MEP pathway is also known as non-mevalonic pathway or mevalonic acid independent pathway. Terpenoids synthesized from acetyl-CoA and pyruvate. Fig. 7.4 isoprene, the building blocks of the terpenes. These compounds are transformed into isobutanol pyrophosphate (IPP) and methyl pyrophosphate by 2 intricate biosynthetic pathways DMAPP. These two molecules IPP and DMAPP are then utilized as the fundamental building blocks for all of the other terpene backbones through the synthesis of granular pyrophosphate (GGP). These two molecules IPP and DMAPP are then utilized as the fundamental building blocks for all of the other terpene backbones through the synthesis of granular pyrophosphate (GGPP). CH3. Fig. 7.5 biosynthesis of terpenes. Activity popular components in commercial insecticides due to their low persistence in the environment and their negligible toxicity to mammals. Phytoesins isolated from a common fern, Polypodium vulgar, and defense against the Phytophagous insect. Ingestion of phytoesins by insects disrupts molting and other developmental process. Many plants such as citrus, mint, basil, peppermint, sage, and eucalyptus contain a mixture of monoterpenes and sesquiterpenes essential oils that impart a characteristic aroma to their foliage. Essential oils have insect repellent properties, repelling potential herbivores even before they take a trial bite. Phenolic compounds. The largest group of secondary metabolites, figure 7.7, in a plant, mostly in edible plants, i.e., vegetables, fruits, and cereals. Since GGPP is a key component of chlorophyll, all plant species contain large amounts of it. Hemiterpenes C5, monoterpenes C10, sesquiterpenes C15, and diterpenes are produced by terpene synthesis, which are main enzymes C20. Volatile terpenes, which are the terpene synthesis only end products and do not undergo any further oxidation, dehydrogenation, acylation, or other processes. The oxidation of organic compounds, such as terpenes, is catalyzed by cytochrome P450. Physiological roles of terpene. Some functions in primary metabolism. Function is herbivore deterrence, can be produced in response to herbivore feeding to attract predatory insects and parasites of the feeding herbivore. Monoterpene esters, known as pyrethroids, occur in leaves and flowers of chrysanthemum species. Insecticidal several structurally different compounds have at least one aromatic hydroxyl substituted ring in common table 7.1. Synthesized by 2 biosynthetic pathways, the shikimic acid pathway and the malonic acid pathway, figure 7.6. Secondary phenolic product biosynthesis starts with the deamination of phenylalanine to cinnamic acid, catalyzed by the enzyme phenylalanine ammonia lyase cal. Cinnamic acid appears to be the precursor for the synthesis of more complex derivatives like lignin, flavonoids, coumarin, and tannin. Fig. 7.6 biosynthesis pathway of phenolic compounds in plants. Class carbon skeleton example C6 simple phenols and benzoquinones. Catechol 2 6 methoxybenzoquinone hydroquinone. Phenol. C6C1 benzoic acid scalic, p-hydroxybenzoic, salicylic, vanillic acid. Highly branched polymer of 3-phenolics alcohol i.e. coniferol, coumaryl, and synapyl. Found in the cell walls of tertiary elements such as trachees and besyl elements. Synthesized from phenylalanine via various cinnamic acid derivatives. Provide mechanical support, defensive role in plants and because of its chemical composition in digestible herbivores. Being bound to cellulose and protein, lignin further reduces its digestibility. Insoluble in water and most organic solvent. Linification restricts the growth of pathogens and is the most common response to infection or wounding. Flavonoids. Largest groups of plant phenolics with a 15C carbon skeleton, comprising 2 benzene rings linked by a 3C bridge are connected through a heterocyclic pyro ring structure, figure 7.8. They fall into 9 main subclasses, including flavonones, flavonols, flavonones, flavanols or catechins, isoflavins, anthocyanidins and proanthocyanidins, and borones. Chalcones and flavonoids with an open C ring are referred to as flavonoids. Soluble in water and accumulate in cell vacuolar compartments. Anthocyanidins, only group of flavonoids that impart color to plants, all other flavonoids are colorless. Anthocyanin, a class of pigmented flavonoids responsible for red, pink, purple, and blue colors to fruits, vegetables, flowers, and grains, especially black soybean, which is an excellent source of anthocyanins. Biological roles of flavonoids. Regarding phytolexins, provide protection against herbivores and pathogens. Controlling the growth and development of plants. Flowers' many colors operate as visual cues to draw insects, birds, and other animals for pollination and seed dissemination. Provide a stress buffer. Anthocyanins serve a number of functions in plants. In addition to acting as pollination and seed dispersal attractants, they also protect plants from UV radiation and provide them antiviral and antibacterial properties. Figure 7.8 Subclassification of flavonoids. Coumarins. The most basic aromatic chemical compounds are coumarin and 7-hydroxycoumarin umbelliferone, which are produced by the reactions of cinnamic acid and p-coumaric acid, respectively, with ring closure. Other coumarin derivatives are esqualane 6, 7-dihydroxycoumarin, pernirin, 7-methoxycoumarin, and sorolin, figure 7.9. Fig. 7.9 A visualization of fluorescent coumarins produced by roots of A star Arabidopsis thaliana COL0 plants. B chemical structures of representative plant-derived simple coumarins and of coumarin iodine. Physiological roles of coumarins. Their role is antimicrobial agents, feeding deterrents, and germination inhibitors. The most prevalent coumarin in flowering plant species, scopolamine, is found in the seed coats and is thought to impede germination. Bergamot oil, which contains the volatile oil coumarin, is used to flavor pipe tobacco, tea, fragrances, fabric conditioner, and other items. Tannins. They are water-soluble and polymerization products of phenolic compounds, responsible for the brown coloration of oak leaves in the fall as well as discoloration of injured plant tissues. Tannins play a defensive role because of their toxic nature, normally due to the potential to bind proteins non-specifically. Tannins are general toxins that significantly reduce growth and affect the survival of many herbivores when ingested. Tannins act as feeding repellents to a great variety of animals. Frequently, immature fruits have very high tannin contents that are mainly concentrated in their outer cell layers, figure 7.9. Fig. 7.9 Types of tannins and their basic structures. Nitrogenous compounds. Many plant secondary metabolites have nitrogen in their structure. Nitrogen-containing compounds include alkaloids and cyanogenic, glycosides, and glucosinolates. Nitrogenous secondary metabolites are synthesized from common amino acids. Alkaloids. Wide class of natural occurring heterocyclic nitrogen-containing organic compounds. Water-soluble and contain at least one nitrogen atom in their chemical structure, figure 7.10. Alkaline in nature alkaloids. Alkaloids include morphine, quinine, ephedrine, reserpine, caffeine, atropine, cocaine, colchicine, and nicotine. Synthesized by plants found in leaf, bark, seeds, and other parts. Act as protection against predators' attacks. First isolated and crystallized alkaloids in 1804 were morphine, a potent alkaloid obtained from Poppy papaver somniferum. Physiological roles. Derived from ornithine, arginine, tyrosine, tryptophan, lysine, and phenylalanine among other common amino acids. Nicotine alkaloid synthesized from nicotinic acid in plants. Morphine is a powerful analgesic or pain reliever. Quinine, obtained from cinchona spp, powerful antimalarial agent, treats arrhythmia. Vincristine and vinblastine are used in the treatment of various types of cancers. Claviceps purpurea reduce uterine hemorrhage after childbirth. Alkaloids. Free alkaloids. N-heterocycles polyamine alkaloids pseudoalkaloids. Cyclopeptidoalkaloids. Atropine palestrine aconidine solanidine. Caffeine. Fig. 7.10 Major alkaloid subgroups and chemical structures of some representatives. Glycosides. It is a glycoside compound that has carbohydrate moiety bound to another functional group by glycosidic bond. This group represents glycone, while the non-sugar group is aglycone or genin, a component of glycoside. Glycone and aglycone linkage, known as glycosidic linkage. Glycosides are classified into 3 groups, i.e. White circle cardiac glycosides are cardinalized, white circle cyanogenic glycosides. White circle saponins. Cardiac glycosides are cardinalides. Naturally occurring glycosides are found in plant species. Active glycosides are the leaves of Deepercurea, foxglove plant, are digitoxin, gitoxin, and betelin. The foxglove plant, which was originally used to treat dropsy and unwelcome buildup of water in the body cavities, is highly regarded as a treatment for congestive heart problems, arrhythmia, and irregular heartbeat to strengthen the heart muscles by increasing the rate of contraction, circulation, and related conditions. Cardinalized glycosides are the milkweeds that produce milky white, cardinalized rich latex. Extremely toxic to higher animals, act as a defense against predators. Cyanogenic glycosides. These glycosides are a toxic class of N-containing secondary metabolites, Fig 7.11. When tissue is damaged, then these glycosides break down and will release HCN. They are an aglycone with a cyanide group attached. Found in the seeds and wilted leaves of various plant species of the Raceae family such as peaches, apples, apricots, cherries, plums, and crab apples. Made by converting amino acids to oxines, which are subsequently glycosylated. In sorghum bicolor, durin is found, which is a major secondary metabolite derived from tyrosine. Cyanogenic glycosides example is amiculin derived from bitter almond and other members of Raceae, which undergoes two-step hydrolysis leading to the release of hydrocyanic acid HCN. The specific fungal pathogen can attack plants while cyanogenic glycosides are produced with formamide hydrolase activity and cyanide resistance. Cyanide inhibits cytochrome C oxidase in the mitochondria and that will block ETS electron transport chain. Fig. 7.11 Cyanogenic glycosides have the following chemical makeup. A. The cyanogenic glycosides general structure, where R is a variable chromatin, which is present in cherry and peach. C. Amygdalin, which can be discovered in the apple, apricot, peach, almond, cherry, and plum kernels. D. The sorghum compound durin, B. Limonaren, which is present in flax seeds, lima beans, and cassava. Saponins. These are chemical molecules with foaming characteristics derived from the silkworth plant Saponaria spp. Saponins are made up of polycyclic aglycones bonded to one or more side chains of carbohydrates. Lipid and water soluble and with a bitter taste. Glycyrrhizin saponin, which is found in licorice Glycyrrhiza glabra, has an expectorant, corticosteroid, and anti-inflammation activities. It is occasionally employed as a sweetener and flavoring addition in foods and cigarettes anti-vegans. Saponins are phytonutrients that can be found in a variety of foods, including vegetables, beans, peas, and soybeans. Some saponins found in foods like oats and spinach, for example, may improve nutrient. Popular in cosmetic formulations such as shampoos, facial cleansers, and cosmetic creams because of their detergent property. Two primary glycosides, steadyoside and ribadioside, are utilized as a natural sweetener in place of normal sugar in many countries. Chapter summary. Secondary metabolites such as terpenoids, phenolics, and alkaloids play a defensive role in plants and also act as modulators in response to abiotic stress. Secondary metabolites lie at the interface between primary metabolism and the interactions of organisms with their external environment. Secondary metabolites are organic compounds produced by bacteria, fungi, or plants, not directly involved in reproduction, development, and normal growth of organisms, in small quantities, so their extraction from the plant is difficult during the stationary phase, also known as idiophase. Broadly classified into terpenes, nitrogenous compounds, alkaloids, flavonoids, and glycosides, etc. Plant stress is a condition in which plant demands are increased due to poor growing conditions. If the amount of stress is too much for the plant, it may cause irreversible damage, shortages in growth, low agricultural yields, or even death. Abiotic factors and biotic factors are the two basic categories into which plant stress factors fall. The biotic factors are the other species that share the environment with the plants and interact with them, whereas the abiotic factors are the various environmental elements such as light, water, and temperature that affect plant growth. Definition. Stress is defined as any environmental condition that prevents the plant from achieving its full genetic potential. OR. Stress is usually defined as an external factor that exerts a disadvantageous influence on the plant. Stress triggers a wide range of plant responses, such as altered gene expression, cellular metabolism changes in growth rates and crop yields. Stress may be broadly categorized into two groups. Figure 8.1. Biotic stress imposed by other organisms. Abiotic stress, the negative impact of non-living factors on the living organism in a specific environment arising from an excess or deficit in the physical or chemical environment. Water deficit, heat stress and heat shock, chilling and freezing, salinity, and oxygen deficiency are major stress factors restricting plant growth. Figure 8.2. 8 stress physiology. Figure 8.1 stress factors in plants. Fig. 8.2 abiotic stress types. Water stress, drought resistance. Fig. 8.3. The ability to maintain tissue hydration and function while dehydrated is the C4 and CA plants' modes of photosynthesis allow them to exploit more arid environments. A plant that is capable of acquiring more water or that has higher water use efficiency will resist drought better. Water deficit can be defined as any water content of a tissue or cell that is below the highest water content exhibited at the most hydrated state. Fig. 8.3 causes of water stress and the responses shown by plants. Fig. 8.4 mechanism of plant reactions to drought resistance. Stress involving water deficit. Water-related stresses can influence plants if the environment contains inadequate water to meet basic needs. Figure 8.4. Environmental conditions that can lead to water deficit are drought, hypersaline conditions, low temperatures, and temporary loss of turgor at midday. Water deficit stimulates leaf abscission. After all the leaves have matured, the total leaf area of a plant, which is calculated as number of leaves x surface area of each leaf, does not remain constant. If plants experience water stress after a sizable leaf area has grown, many drought deciduous desert plants drop all of their leaves during a drought and sprout new ones following rain, at which point the leaf will eventually senesce and fall off. Adaptive response to water deficit. Decreased leaf area. Water deficit improves water extension into deeper, moist soil. It increases resistance to liquid phase water flow. It also increases wax deposition on the leaf surface. It alters energy dissipation from leaves. Osmotic stress induces a CAM cycle in some plants. Stress responsive genes are regulated by OB-dependent and OB-independent processes. Fig. 8.5. Fig. 8.5 stress responsive genes and their regulation by OB-dependent and OB-independent processes. Osmotic adjustment of cells helps maintain plant water balance. Plants continue absorbing water only if their water potential, psi W, is lower, more negative than that of the soil water. Osmotic adjustment, a method that allows water potential to be lowered without also lowering turgor or cell volume. Psi W equals psi S plus psi P. Changes in solute potential, psi S, the osmotic component of water, psi W, are what cause a change in the cell's water potential. Osmotic adjustment is defined as a net rise in solute content per cell that is unrelated to changes in cell volume brought on by the loss of water. Compatible solutes, osmolytes. Mostly neutrally charged at physiological pH, even non-ionic or zwitterionic and are excluded from hydration shells of macromolecules. Compatible solutes proline, glycine butane, beta-alanine butane, choline sulfate, mannitol, and phenol contribute to osmotic adjustments. Salt accumulation in soils impairs plant function and soil structure. High concentration of NAL plus IE, sodicity and high concentration of total salts IE, salinity can not only injure plants directly but also degrade the soil structure and also decreases the porosity and permeability of water. Salt injury involves both osmotic effects and specific ion effects. Low osmotic potential is generated by dissolved solutes in the rooting zone, lowers the soil water potential. So, a downward gradient of water occurs to maintain water potential between the soil and the leaves. Heat stress and heat shock. Heat shock may arise in leaves when transpiration is insufficient or when stomata are partially or fully closed, and radiance is higher. May arise in germinating seedlings when the soil is warmed by the sun. Even may arise in organs with reduced capacity for transpiration, e.g., fruits. It is known as induced thermodulance when regular, brief exposure to sublethal heat shocks leads to tolerance to temperatures that would otherwise be lethal. At high temperatures, photosynthesis is inhibited before respiration. Both photosynthesis and respiration are reduced at high temperatures. However, as the temperature rises, photosynthetic rates decline before respiratory rates do. The temperature compensation point is the point at which the quantity of CO2 fixed by photosynthesis matches the amount of CO2 emitted by respiration in a particular period of time. When the temperature rises past the temperature compensation point, photosynthesis is unable to take the place of the carbon that is used as a respiration substrate. Fruits and vegetables lose their sweetness as a result, and carbohydrate reserves decrease. High temperature reduces membrane stability. In oleander, Miriam oleander, adaptation to high temperatures is linked to a higher level of fatty acid saturation in membrane lipids, which reduces the fluidity of the membrane. In the aqueous phase of the membrane, the strength of hydrogen bonds and electrostatic interactions between polar protein groups decreases at high temperatures. High temperatures can change the structure and composition of membranes and result in ion leakage. Processes like photosynthesis and respiration that rely on the activity of membrane-associated electron carriers and enzymes are inhibited by membrane disruption. Heat shock proteins. The typical response to heat stress is a decrease in the synthesis of normal proteins, accompanied by an accelerated transcription and translation of new proteins known as heat shock proteins, HSPs. Table 1. Plants respond to abrupt temperature increases of 5 to 100 C by producing heat shock proteins, a unique set of proteins. Many cell proteins that serve as enzymes or structural elements get unfolded or misfolded in response to heat stress, which results in the loss of normal enzyme structure and activity. Such improperly folded protein frequently precipitates in clumps, posing major issues for the cell. HSPs function as molecular chaperones and help misfolded, aggregated proteins fold properly and avoid misfolding altogether. This enables proper cell function under high stress conditions. Class size KDA, location HSP 100,114 cytoplasm. Class size KDA, location HSP 100,114 cytoplasm, HSP 908094 cytoplasm, or HSP 706971 cytoplasm, mitochondria HSP 6010 chloroplast CHL MITSM HSP 15 to 30 cytoplasm, MIT, chloroplast table 8.1 classes of heat shock proteins, HSPs. A transcription factor mediates HSP accumulation in response to heat shock. HSPs are discovered in fruit fly, Drosophila melanogaster, and appear to be ubiquitous in plants, animals, fungi, and microorganisms. Heat shock response appears to be mediated by one or more signal transduction pathways, one of which involves a specific set of the transcription factor called heat shock factors that regulate the transcription of HSP mRNAs. HSP 70s bind and release misfolded proteins. HSP 60s that produce huge barrel-like complexes that are used as chambers for protein folding. HSP 101s that mediate the disaggregation of protein aggregates, and SHSPs and other HSPs that bind and stabilize different complexes and membranes. Figure 8.6. Diagram source cities and Zeiger. Fig. 8.6 The heat shock factor, HSF, cycle activates the synthesis of heat shock protein mRNAs. Numerous other proteins have been identified that act in a similar fashion to stabilize proteins and membranes during dehydration, temperature extremes, and ion imbalance. These include LEAT DHN, RAB protein family. Late embryogenesis abundant proteins accumulate in response to dehydration during the latter stages of seed maturation. Most leaf proteins belong to a more widespread group of proteins called hydrophilins, have strong attraction to water. DHNs dehydrins accumulate in plant tissue in response to a variety of abiotic stresses. Dehydrins, like leaf proteins, are highly hydrophilic and intrinsically disordered proteins. Their ability to serve as molecular shields and as cryoprotectants has been attributed to their flexibility and minimal secondary structure. Since both lias and DHNs are often induced by ABA, they are sometimes referred to as RABs responsive to ABA. Chilling and freezing. Temperatures below freezing are insufficient for normal growth but not for ice to form. Usually, organisms from the tropics and subtropics are vulnerable to cold harm. Chilling injury happens when plants that are used to relatively high temperatures, 25 to 35 degrees Celsius, are dropped to 10 to 15 degrees Celsius. Growth is halted, lesions or discoloration appear on leaves, and the foliage seems soggy as if it has been submerged in water for a long period. Plants may wilt if their roots become chilled. Species that are often susceptible to chilling might exhibit noticeable variety in how they react to cold temperatures. Chiller resistance is enhanced by genetic adaptation to the lower temperatures associated with high altitude. Chiller resistance is enhanced by genetic adaptation to the cooler temperatures associated with high altitude. Cold shock, or sudden exposure to temperatures around 0 C, raises the risk of harm. On the other side, freezing injury happens when the temperature is below the freezing point of water. As with chilling, full induction of tolerance to freezing necessitates a period of acclimatization at low temperatures. Membrane properties change in response to chilling injury. Inflicted by cooling, plants' leaves exhibit decreased respiration rates, delayed glucose translocation, decreased photosynthesis, decreased protein synthesis, and increased protein breakdown. Loss of solutes to the water is a sign of damage to the tonoplast and possibly the plasma membrane. In turn, a decrease in photosynthesis and respiration is a sign of damage to the mitochondrial and chloroplast membranes. The activities of the integral membrane proteins, including H+ acases, carriers, and channel-forming proteins that control the transport of ions and other solutes as well as the transport of enzymes necessary for metabolism are significantly influenced by the physical characteristics of the lipids. In addition, chilling-sensitive leaves subjected to high photon fluxes and chilling temperatures are photoinhibited, producing acute damage to the photosynthetic apparatus. As the membranes become less fluid, their protein constituents are unable to function normally. When plants become acclimated to chilling temperatures, the activity of desaturase enzymes increases and the fraction of unsaturated lipids grow. This adjustment lowers the temperature at which the membrane lipids start a progressive phase shift from fluid to semicrystalline and enables membranes to remain fluid at lower temperatures. During freezing stress, extracellular ice crystal formation causes cell hydration. Plants subjected to freezing temperatures must contend with the formation of ice crystals, either extracellularly or intracellularly. Intracellular ice crystal formation nearly always proves lethal to the cell. However, the water in the apoplast is relatively dilute and therefore has a higher freezing point than that of a more concentrated symplast. As a result, ice crystals tend to form in the apoplast and in the xylem tracheids and vessels, along which ice can quickly propagate. The formation of ice crystals lowers the apoplastic water potential, psi, which becomes more negative than that of the symplast. Unfrozen water within the cell moves down this gradient out of the cell toward the ice crystals in the intercellular spaces. As water leaves the cell, the plasma membrane contracts and pulls away from the cell wall. During this process, the plasma membrane, rigidified by the low temperature, may become damaged. More water flows down the gradient toward the frozen water as the temperature drops. Limitation of ice formation contributes to freezing tolerance. Several specific plant proteins may inhibit the development of ice crystals via an uncollated process, i.e., a result that is independent of solutes' ability to reduce the freezing point of water. Cold temperatures trigger the production of these antifreeze proteins, which bind to the surfaces of ice crystals to stop or inhibit crystal development. Soluble sugars build up in the cell walls of winter cereals during cold acclimation, which may help limit the development of ice. From the leaves of cabbage that have been exposed to cold, a cryoprotected glycoprotein has been found, Brassica oleracea. The protein shields non-acclimatized spinach, Spinacia oleracea, thylakoids from damage from freezing and thawing in vitro. ABA and protein synthesis are involved in acclimation to freezing. As a result of changes in gene expression, multiple different proteins accumulate during acclimatization to cold, which is important for the development of freezing tolerance. The RAB DHN protein family, responsive to ABA, late embryo abundant, and dehydrin, respectively, and several of the proteins that are activated by low temperature share similarity according to the isolation of the genes for these proteins. ABA seems to contribute to the development of cold tolerance. Winter wheat, rye, spinach, and Arabidopsis thaliana are all cold-tolerant plants, and their freezing tolerance also rises as they become toughened by water shortages. A modest water deficit or low temperatures, either of which boosts endogenous ABA concentrations in leaves, both of which increase this tolerance to freezing. Numerous genes are induced during cold acclimation. While, housekeeping, protein synthesis is not significantly downregulated during cold episodes, proteins produced in the absence of stress, it is essentially shut off during heat stress. Cold stress increases the production of numerous heat shock proteins that can operate as molecular chaperones in the same way as heat stress does. Antifreeze proteins are another significant family of proteins whose expression is increased by cold stress. These proteins can limit ice crystal development in an uncollated manner, reducing freeze damage at intermediate freezing temperatures. Antifreeze proteins, also known as thermal hysteresis proteins, provide aqueous solutions with a feature of thermal hysteresis, which promotes the transition from liquid to solid at a lower temperature than the transition from solid to liquid THPs. Cold stress also causes an upregulation of a different set of proteins linked to osmotic stress. These proteins include the lead proteins, proteins for membrane stabilization, and proteins involved in the manufacture of osmolytes. It was discovered that all antifreeze proteins belong to a class of proteins called endokinases and endoglucanases, which are triggered during infection by various pathogens when the specific genes encoding for these proteins were cloned and sequenced. Pathogenesis-related PR proteins are a class of proteins that are hypothesized to protect plants against pathogens. Salinity stress. Due to the increased use of subpar water for irrigation and soil salinization, salinity is a significant abiotic factor that restricts plant development and productivity in many parts of the world. Complex physiological features, metabolic processes, and molecular or gene networks are required for plant adaptation or resistance to salinity stress. Pure water as vapor is removed from the soil via evaporation and transpiration, which concentrates solutes there. When there is no way to flush accumulated salts into a drainage system and irrigation water has a high solute content, salt concentrations can rise quickly to levels that are harmful to salt-sensitive species. Because high salt concentrations in the soil and plants cause osmotic stress during the early stages of salinity stress, which is also known as hypersmotic stress, the ability of root systems to absorb water declines and water loss from leaves is increased. So does satiety, and high concentrations of total salts, referred to as salinity. In some regions, in addition to NaCl, Ca2+, Mg2+, and so forth, can also significantly contribute to saltiness. In addition to directly harming plants, sodic soils high NaCl concentration can deteriorate the soil structure, reducing porosity and water permeability. Salinity depresses growth and photosynthesis in sensitive species. In general, two kinds of plants can be distinguished depending on how they react to salt concentrations that are high. Saline soils are home to halophytes, which complete their life cycles there. Diagram source: Tees and Zeiger. Figure 8.7. The growth of different species subjected to salinity relative to that of unsalinized controls. Salt accumulation in soils impairs plant function and soil structure. While studying the effects of salts in the soil, we distinguish between high concentrations of NaCl+, referred to as non-halophytes, often known as glycophytes literally, sweet plants, are less able to withstand salts than halophytes. Usually, glycophytes exhibit symptoms of growth inhibition, leaf browning, and loss of dry weight over a certain salt concentration. Salt injury involves both osmotic effects and specific ion effects. The rooting zones dissolve solutes produce a low, more negative osmotic potential that reduces the soil's water potential. A finite amount of water that the plant can extract from the soil profile during soil desiccation results in ever-declining water potentials. In the majority of saline situations, there is a substantial, almost infinite supply of water with a steady, low water potential. The majority of plants can osmotically adapt to growing in salty soils. Reduced water potential is produced while preventing turgor loss, which would limit cell growth. Specific ion toxicity effects also arise when harmful quantities of ions, particularly Na+, Cl, or SO4 build in cells, in addition to the plant reactions to low water potential. Higher plant cells have cytosols that are 100 to 200 K+ and 1 to 10 Na+ under non-saline circumstances, which is an ionic milieu where many enzymes perform best. At high concentrations, Na+ can displace Ca2+ from cotton root hair plasma membranes, causing a change in plasma membrane permeability that can be seen as leakage of K+ from the cells. An abnormally high ratio of Na+ to K+ and high concentrations of total salts inactivate enzymes and inhibit protein synthesis. When excessive quantities of Na+ and or Cl accumulate in chloroplast, photosynthesis is hampered. Plants use different strategies to avoid salt injury. Salinity stress is the accumulation of NAD+ and Cl- ions in tissues of plants exposed to soils with high NAD+ concentrations. Entry of both NAD+ and Cl- into the cells causes severe ion imbalance, and excess uptake might cause significant physiological disorders. High NAD+ concentration inhibits the uptake of K+ ions, which is an essential element for growth and development that results in lower productivity and may even lead to death. Plants attenuate salt injury by excluding salt from merostems, particularly in shoot leaves which are actively expanding and photosynthesizing. In salt-sensitive plants, resistance to moderate levels of salinity in soil depends on the ability of roots to prevent harmful ions from reaching the shoots. The casparian strip creates a restriction to the movements of ions into the xylem. So, to bypass the strips, ions need to move from the apoplast to the symplastic pathway. This offers salt-resistant plants a mechanism to exclude harmful ions. NAD+ ions enter roots passively by moving down an electrochemical potential gradient, so root cells must use energy to extrude NAD+ actively back to the outside solution. Opposite this, Cl is excluded by negative electric potential across the cell membrane. Movement of NAD+ into leaves is further minimized by absorption of NAD+ from the transpiration stream during its movement from roots to shoots and leaves. Oxygen deficiency. Oxygen deficiency in the rooting zone occurs with poor drainage after rain or irrigation, causing depressed growth and yield of dry land species, in contrast with native wetland vegetation that tolerates such conditions. Plants under natural or experimental conditions can be subject to a range of O2 concentrations from normal normoxia through deficient hypoxia to total absence anoxia. Hypoxia occurs when the oxygen level limits aerobic respiration, usually between 1% and 5%, while anoxia takes place when oxygen is absent in the environment. Flooding-sensitive plants are severely damaged by 24 hours of anoxia. Other plants, particularly species not adapted to grow in continually wet conditions, and many crop plants are affected by flooding in a milder way, considered flooding-tolerant plants. Anaerobic microorganisms are active in water-saturated soils. When soil is completely depleted of molecular O2, the function of soil microbes becomes significant for plant life and growth. Anaerobes derive their energy from reduction of nitrite to nitrite or nitrous oxide and molecular nitrogen. These gases are lost to the atmosphere in a process called denitrification. As conditions become more reducing, anaerobes reduce Fe3+ to Fe2+, because of its greater solubility. Fe2+ can raise to toxic concentrations when some soils are anaerobic for many weeks. It's possible for other anaerobes to convert the respiratory toxin hydrogen sulfide H2S from the sulfate SO42. Acetic acid and butyric acid are two bacterial metabolites that are released into the soil water when anaerobes have an abundant supply of organic substrate. These acids, together with reduced sulfur compounds, are responsible for the foul smell of wet soil. When present in high concentrations, each of these compounds produced by bacteria in anaerobic environments is poisonous to plants. Damaged O2 deficient roots injure shoots. A drop in leaf water potential and wilting are frequently caused by the reduced water permeability of roots, albeit this decrease is only transitory if the stoma is closed, stopping additional water loss through transpiration. Lack of oxygen in the roots, along with a lack of water or high salt content, can promote the creation of ABA and the transfer of ABA to the leaves. However, the older, lower leaves increased synthesis of ABA is primarily responsible for the stomatal closure under these circumstances. These wilting leaves transmit ABA to the younger, turgid leaves, which causes stomatal closure. Most plant tissues cannot tolerate anaerobic conditions. The majority of higher plant tissues cannot endure extended anaerobic conditions. Root tips of maize, for example, stay viable for only 20 to 24 hours if they are suddenly deprived of O2. Highly reactive oxygen species are produced during aerobic metabolism, and they are often detoxified by cellular defense systems that involve superoxide dismutase SOD. Superoxide radicals are changed by this enzyme into hydrogen peroxide, which is then changed into water by peroxidase. SOD may be able to handle the increase in O2 that happens when the leaves emerge into the air from water or mud in the tolerant species, which could help them resist post-anoxic stress. Acclimation to O2 deficit involves synthesis of anaerobic stress proteins. These glycolytic and fermentation pathway enzymes have been identified as the majority of these anaerobic stress proteins. An increase in intracellular Ca2+ is one of the first processes to happen after the reduction of O2. Evidence suggests that the anoxia signal is transduced via this calcium signal. ADH and sucrose synthase mRNA levels rise in cultured maize cells within minutes of the commencement of anoxia as a result of a rise in the cytosolic Ca2+ concentration acting as a signal. Chemicals that prevent an increase in intracellular Ca2+ concentration also prevent anoxia from inducing the expression of the genes for ADH and sucrose synthase and significantly increase the susceptibility of maize seedlings to anoxia. Changes in the rate of transcription of the anaerobic stress genes cause their mRNA to accumulate. Biotic stress. Biotic stress in plants occurs due to biological factors, i.e., insects and disease, and plant responds to these conditions with alternation in the chemical and physical characteristics of cell walls and synthesis of secondary metabolites. These responses, which are collectively known as hypersensitive reactions, isolate potentially pathogenic organisms, also stop their development, spread across the plant. Hypersensitive reactions include white circle stimulation of defense-related genes and also the formation of their products, pathogenesis-related PR proteins, figure 8.8. White circle induction of genes that code for enzymes responsible for the synthesis of antimicrobial compounds such as phytolexins and isoflavonoids that limit the growth of pathogens. White circle alterations in cell wall composition are accompanied by its enhanced strength due to the accumulation of lignin, callose, and superin in cell walls alongside hydroxyproline-rich lipoproteins. White circle invaded cells trigger programmed cell death (PCD), which leads to the formation of necrotic spots at the site of infection. Fig. 8.8 Biotic stress and plant responses. Arbuscular mycorrhizal associations and nitrogen-fixing symbionts include closely related signaling mechanisms. In leguminous plants, Symark and many other genes are responsible both for legume modulation and for arbuscular mycorrhizal formation. Key symbiotic signals, not factors, which are produced by rhizobia. The arbuscular mycorrhizal fungus releases lipid oligosaccharides called MYC factors, stimulate the formation of mycorrhiza in several plants. Rhizobia can increase nutrient availability, stimulate root branching, and protect against pathogens. Fig. 8.9. Plant growth promoting rhizobacteria (PGPR) provides several beneficial services to growing plants. PGPR microbes can provide cross protection against pathogenic organisms by activation of induced systemic resistance pathway and also control the buildup of harmful soil organisms. Fig. 8.9 Protection of plants with PGPR. Pathogen invasion response or plant defenses against pathogens. Table 8.1. Plant pathogens penetrate through cell walls by secreting lytic enzymes, as well as through stomata, hydathodes, lenticels, and wound sites caused by insect herbivores. Phloem feeders. All plants have pattern recognition receptors (PRRs) or microbe-associated molecular patterns (MAMPs). Pathogen infection can produce molecular warning signals that are recognized by cell surface pattern recognition receptors. In response to infection, many plants produce phytolexins, secondary metabolites with strong antimicrobial activity. A plant that survives local pathogen infection often develops increased resistance to subsequent attack, a phenomenon called systemic acquired resistance (SAR). Interactions with non-pathogenic bacteria can stimulate induced systemic resistance (ISR). Hypersensitive response (HR) within an hour and inhibit infection to other areas by PCD of infected tissue. Table 1. Table 8.1 Responses to pathogen invasion or plant defenses against pathogens. Fig. 8 Saudi reals and 10 signaling during the response to pathogens. White circle systemic acquired resistance. White circle induced systemic resistance mechanism of SARS systemic acquired resistance. Viral infection of tobacco induced accumulation of a distinct set of proteins. Pathogenesis-related proteins. PR proteins. Figure 8.11. 9 gene families induced in uninfected inoculated plant leaves, known as SAR genes, have a direct antimicrobial activity or related to antimicrobial proteins classes. SAR signaling during the response to pathogens. The resistance that developed in the distal, untreated portion of pathogen inoculated plants is termed systemic acquired resistance. Induced resistance, an increased expression of natural defense mechanisms of plants against different pathogens provoked by external factors. Forms of induced resistance. White circle localized acquired resistance. Morphological and biochemical changes of SAR-protected plants have a faster lionification response, corresponding with an increase in peroxidase activity. Other changes like increased glucose, fructose concentration in systemic tissue, induction of lipoxygenase, phytolexins, and hydroxyproline-rich lipoprotein. Salicylic acid is not a translocated signal responsible for ISAR but is required in signal transduction. Two pathways in plants. White circle NPR1, non-expressor of PR genes. Dependent figure 8.11. White circle NPR1 independent SAR pathway. Figure 8.12. In the systemic defense signaling network, SNI1 suppressor of 1 Nepalese rupee is a negative regulator of SAR. MEPK4, a positive regulator of ISR and a negative regulator of SAR. Fig. 8.11 Pathogenesis-related proteins. NPR1 is a transcriptional coregulator, acts downstream of SAW, and plays an essential role in SAW-dependent defense signaling pathways. Fig. 8 Saudi reals and 12 halalas. The whole plant. Resistance response. Phytolexins. Phytolexins are a diverse group of secondary metabolites or antimicrobial compounds synthesized around the infected site. Phytolexins require transcriptional or translational activity in the plant once the pathogen has been detected. Various sesquiterpenes are produced as phytolexins. Fig 8.13. Fig. 8.13 examples of phytolexin structures that are antibacterial. A. Scopolamine from tobacco. B. Camelexin. From Italiana, C. Saccharinidin, D. Nonylactone B. From rice, and E. Glucosinolates from Brassicacea are only a few examples of the compounds. For Arabidopsis tryptophan and methionine-derived glucosinolates are represented by the structures of the R groups of indole 3-O-methyl E, left, and 4-methylsulfonyl-butylglucosinolate E, right, respectively. Proteins and factors in plant pathogen interaction. Plant innate immunity includes effector-triggered immunity (ETI) and camp-triggered immunity (PTI). Figure 8.14. Camp-triggered immunity (PTI) and effector-triggered immunity are activated as a result of apoplastic pathogen-associated molecular patterns. Camps, apoplastic effectors, intracellular effectors, or modified effector targets being actively recognized by receptors in the plasma membrane or resistance R proteins in the cytoplasm (ETI). The co-perception of pathogen-derived components is mediated by helper proteins and guard proteins decoys. Pathogens utilize host proteins S proteins produced by plant susceptibility genes S genes to assist entry and proliferation, leading to host compatibility. Class 1, Class 2, or Class 3 S proteins include MLO, CESA3, and sweets. White circle Class 1 genes provide features that facilitate the entrance of a pathogen. White circle Class 2 genes increase innate immunity when the gene is disabled, and white circle Class 3 genes encode substrates essential for the pathogen. Figure 8.14 Plant innate immunity, camp-triggered immunity (PTI), and effector-triggered immunity (ETI). Chapter summary. Plant stress factors are mainly categorized into two main groups: abiotic factors and biotic factors. The abiotic factors include different environmental factors that affect plant growth, such as light, water, and temperature, while biotic factors are the other organisms that share the environment and interact with the plants. Stress may be broadly categorized into two groups: biotic stress and abiotic stress. Abiotic stress includes water deficit, heat stress and heat shock, chilling and freezing, salinity, and oxygen deficiency.